Today's presentation is Found in Translation, Building an Early Development Strategy for Complex Biologics. Our speakers are Christina Mayer and Aaron Moss. Christina is currently an Associate Director of Clinical Pharmacology in the Integrated Drug Development Group at Certara Strategic Consulting. Dr. Mayer has expertise in non-clinical and translational and early to mid-stage clinical development of oncology biologic drugs. She earned her Bachelor's of Science degree from Boston College and her PharmD from UNC Chapel Hill. She then went on to pursue a postdoctoral fellowship in clinical PK/PD at UNC Chapel Hill and GSK. Prior to coming to Certara, Christina worked as a clinical pharmacology lead at Janssen Biotherapeutics. Joining Christina today is Dr. Aaron Moss. Aaron is currently a director in the Integrated Drug Development Group at Certara Strategic Consulting. He brings to Certara over a decade of experience in translational modeling and simulation. At Certara, Aaron supports the progression of lead candidates from discovery and early clinical development through to regulatory submission. He holds a PhD in pharmaceutics from the University of Washington and is also an affiliate instructor in the School of Pharmacy in the Department of Pharmaceutics at the University of Washington. Christina and Aaron, welcome to today's event. I'll now turn it over to Christina to begin the presentation.
Thanks, Suzanne. So today's topic is building an early development strategy for complex biologics. And first, we'd like to set the stage by describing biologics and their growing complexity, then discuss the biologics roadmap from new molecular entity or NME declaration to IND or investigational new drug submission, including key milestone activities like developing the target product profile, TPP, evaluating first-in-human risk, and selecting the first-in-human starting dose. Lastly, we will describe clinical pharmacology applications in the early development of biologics, namely phase 1 and 1B up to phase 2, that complement and follow the NME to IND development process. So let's start with the basics. What is a biologic? Biologic therapeutics, which we'll refer to in this presentation simply as biologics, are drugs isolated from natural sources, whether that be human, animal, or microorganism. Examples include proteins, nucleic acids, viruses, or cells. And biologic drug products are usually complex mixtures produced by biotechnology, not as easily characterizable or manufactured as small molecules. They're typically administered parenterally to bypass degradation in the GI tract. And in the U.S., biologics are regulated by the FDA and may be reviewed by both the Center for Biologics Evaluation and Research or the Center for Drug Evaluation and Research. One question I'm asked frequently is, how big are biologics? One traditional classification of drugs is by molecular weight, where they fall into the category of either small or large molecules, where a molecular weight cutoff of around 1 kilodalton typically distinguishes the two. Large molecule biologics are typically thought of as traditional monoclonal antibodies, or MAB therapeutics, like the IgG4-based nivolumab at 146 kilodaltons. But they could also be much smaller than MABs. They could be smaller protein therapeutics like epoetin at 30 kilodaltons or the MAB fragment blinatumomab at 54 kilodaltons. They can be even smaller platforms like small interfering RNA like patisiran at 14 kilodaltons. Biologics can also be much larger than traditional MABs. They could also be antibody-drug conjugates with a linker and a payload for a slightly higher molecular weight. For example, brentuximab vedotin at 153 kilodaltons. They could be viruses like herpes simplex virus in PVEC or adeno-associated virus in voretigene neparvovec. Now we're getting up to 100 to 200 nanometers. Then they can be even as large as human T cells in CAR T therapy, such as tisagenlecleucel, which can be in the range of 5 to 10 microns. So biologics, defined as being produced from a living organism, can be a wide range of sizes. And why does this size matter? Size matters because it impacts where the drug can go, how it can exert its desired effect, how it can exert undesired effects, and how it can be quantified and characterized. But size isn't the only important factor. We now have entered an era of sophisticated engineering and formulation of biologics in an attempt to target complex disease biology, particularly in areas like immuno-oncology, which Dr. Moss will now describe further.
Thank you, Christina. Obviously, while they also encompass a wide range of sizes, biologics these days encompass a wide array of mechanisms and complexities. And clearly, antibodies were the first mainstream biologic for obvious reason. This was because of its nature, nature's drug, and it was effective for binding and blocking receptors with specificity and generally with a lot of safety. Click. You advance the slide, please. Protein engineering advances and the interest in modified distribution pharmacokinetics led to using antibody fragments as opposed to simply using antibodies. It wasn't long before modulation and specifically stimulation of our immune system became possible. And immune agonists like TGN-1412, which we refer to as the CD28 supermab, were being tested for oncology, often with scary results. Bispecific T-cell engagers took even more advantage of our potential to modify the immune system by directly bridging activated T-cells with the target tumor cells. Then we decided maybe we should just directly inject T-cells that we've taught to interact with the target tumor instead of going through the process of building a drug. And ultimately, in the scale of complexity, really we can encompass any sort of modality where now viruses and other devices and carriers are used to deliver genes to immune cells. And these cells now are the drug development device. Regardless of the complexity or the size, all therapeutics must achieve certain milestones in development in order to ensure the optimal candidates progress, while the less optimal do not. The only way that this can occur is through informed decision-making. A few of the primary milestones that are important for consideration in development are NME declaration, or new molecular entity declaration, something that's not listed here, but lead selection, opportunities to meet with health authorities at the pre-IND phase, and then filing an investigational new drug application or an IND. Now, obviously, these are only describing preclinical events, but these are critical at the early phases of development and at the inception of the program to be considering these. The questions of what data is required to move your therapeutic through these milestones, how are you going to collect that data, and when do you need it, are not only important for meeting the milestones, but also for your assay development strategy and your first-in-human dose selection strategy. In general, there are a few major pillars to consider through these development milestones, and we're going to step through them with some explanations, definitions, and brief examples of how you might consider them. Obviously, these all in themselves are topics that could be webinars, so we're not going to be able to approach the level of detail you might want to. For example, we're not going to go into modeling and simulation guidance today, but there are also webinars available on this topic here. The important takeaway here is that there is not one correct method to carry a program through preclinical development into first-in-human, but regardless of the specific methodologies, often these considerations are going to be consistent. And so what are those considerations, and how do we develop tools to approach those? Usually the common questions about preclinical development involve things like, what do we do in the absence of certain data or models? And much like painting a picture, the context of the body of data, when integrated, is what will point to the answer. Around the TPP, or the target product profile, are examples of topics and decisions that will be addressed at some point in development. And while they may not at first glance appear to be related, they are in fact inseparable. Because of this, integration of all of the data, information, and experience is not only valuable, it is required. And throughout the development, the TPP as a living document should be readdressed in light of the growing information being integrated. So what is a TPP? Well, it's a dynamic resource that serves to guide development decisions across functional areas by creating alignment around attributes and outcomes. What it is not is a static document or a table dictated by any one functional area to direct the development of a therapeutic according to what they deem is important. The format of this document can take many different versions, and how this is created is really up to the organization that's developing it and the molecule and therapeutic plan. Be sure to include all of the relevant information that you can and ask the questions that you may not know answers to, but those that will be important in the future. And remember that this is a living document and needs to change when necessary, or else it will not be relevant with the integration of the data at hand. And so once you have defined the molecule in terms of its fundamentals and you have understood in general where you plan this molecule to go, one of the most important things is assessing the risk. And so now I'm going to hand it off to Christina.
Thanks, Aaron. So the goal of a first-in-human risk assessment is to understand potential safety concerns related to administration of your therapeutic and to help guide the first-in-human starting dose selection strategy. So first-in-human risk categories have been proposed in guidances by regulators, and this is a list here that has been adapted from an EMA guidance. It includes considerations as follows. Is any component of the therapeutic novel? Is it a novel target or a novel platform? Does the biological mechanism have potential for downstream effects for which the magnitude should be considered? For example, cytokine release syndrome. Is there a cross-binding to similar targets? Is the translational animal model relevant? And if so, in what aspects is it relevant and in what aspects is it not relevant? For example, perhaps expression in a non-human primate is much lower compared with humans, and that will need to be considered in the translation. What are the pharmacodynamic characteristics and what is the relationship to PK? For example, is there a steep dose safety relationship or are there irreversible effects? How relevant and specific are the inputs we're using for calculation of first-in-human starting dose? And how accurate is our human PK projection based off the target-mediated drug disposition we may see in a non-human primate? How reliable are our Mabel assays? So all of these questions go into our risk assessment and more. And by answering these questions for our compound, we can then get a sense of the overall risk, low, moderate, or high, and then implement strategies to mitigate that risk in both dose selection and study design. For example, if our biologic therapeutic is a novel platform like a tetravalent bispecific antibody, which acts as an immune agonist against a novel target, the first-in-human risk would be considered high, and a Mabel dosing approach, which Aaron will explain further, using the most sensitive and specific in vitro assay would likely be utilized. This resulting starting dose could be 4 logs lower than the predicted optimal biologic dose, so maybe single patient sentinel cohorts would be incorporated with cohort by cohort analysis and a plan to expand to 3+3 design upon reaching the predicted target PK/PD. So we consider all of the risks around the compound in order to both dose select and design our first-in-human study. And a deep understanding of the molecular engineering, biology, toxicology, pharmacology, and translational science is required to properly evaluate this risk in a first-in-human setting. So I've given you one example, but what are the many ways we can select a biologic first-in-human starting dose, and what considerations go into this critical decision? I'll hand it off to Aaron to explain further.
Thanks, Christina. Selecting the first-in-human starting dose is really one of the truly difficult aspects of preclinical development, and for obvious reasons. While there are many challenges involved, a couple of the common challenges immediately come to mind are here. Complex biologics, especially those that involve modulation of the immune system, can have the potential, as you know, to cause exaggerated and unexpected pharmacology. And another common challenge is that preclinical models often fail to predict the potential for human toxicities, but also they may underestimate or fail to predict the beneficial immune modulation. So in the light of these challenges, how do we provide dosing guidance for molecules either with high risk characteristics or with minimal translatable data? This is a very complicated slide, but I want to start with a few basics to orient you. And let's start simply with the upper right-hand corner. In general, we want to start with risk. define the level of risk associated with the molecule. In general, higher risk molecules are going to have lower starting doses, and hopefully this is intuitive as to why. Finally, when you've arrived at your level of risk, try to understand and define what your dose or exposure response will be, and utilize some of the relevant pharmacodynamics or even tox parameters depending on that level of risk. And looking at this figure below, a very common figure, starting on the x-axis, you see the dose or exposure, and on the y-axis, you're seeing effect. And as you travel upward in terms of dose or exposure, we will always see pharmacology before we see therapeutic events or toxic events. So understanding where you are in the dose or exposure response relationship will help you understand where your first-in-human dose should be. And I've listed some of the major kinds of dose selection methods here, ranging from the highest risk molecules, where you might use a Mabel or a minimum anticipated biological effect level, a modified Mabel approach, which might use selecting a target receptor occupancy, minimal pharmacologically active dose or minimally effective dose, which is based on pharmacology parameters, or a simple tox parameter such as NO8EL, or a no observed adverse event level, or highest non-severely toxic dose scaled to a human effective dose. And so all of these are methods that go along with the level of risk. And then finally, use more than one method to calculate to obtain multiple potential starting doses, and be clear about the rationale and why you are choosing those. Let's go through a couple of examples. Let's start with low-risk scenario and a non-oncology drug. Now, we've already identified that this is a low-risk molecule, so in general, you're going to consider a higher starting dose just based on principle. In this case, this might require a no observed adverse event level, and the reason being that the novelty of the molecule and the observed mechanism may not justify using such a low starting dose as a Mabel. Here, one of the important considerations is that you must have an appropriate tox species and have the appropriate justification for that species. And you will go about converting your NO8EL of milligrams per kilogram dose to a human equivalent dose, and you might apply a safety factor. In order to justify your species as a relevant species, here's some things to consider. What is the sequence homology, and how similar is that species to human? How similar is the biology between that species and human, and can you make translatable arguments for using it? What about the binding and the affinity? Are they similar between that target species and human, and how similar are the target proteins? In general, ADME should be translatable from cyno to human using established methods. And you have to consider that there may be differences in target expression and Fc gamma receptors and the binding between cyno and human. And now let's just take a simple high-risk scenario. So here is a molecule that you have previously considered having a higher risk for your first-in-human starting dose, whether it be a cytokine release, a novel mechanism of action, or just difficulty in understanding how to calculate a first-in-human dose. In this situation, hopefully you understand now that you would clearly use a Mabel approach primarily. And an in vitro calculated Mabel is most relevant for dose selection. And here, one of the important considerations, which we'll go into further, is which assays are most relevant for this drug using a Mabel approach. Something to consider, and an extremely helpful exercise that has multiple benefits for use of Mabel or modified Mabel-based molecules, is creating and maintaining a master table of key pharmacological parameters. And I'm showing an example of what such a table might look like here. This will be potentially used in the calculations, but also it serves as a repository of this information that's easy to read and concise for the entire project team to understand, because not everybody is a pharmacology expert. And also, it can produce meaningful comparisons with comparator molecules, which I strongly recommend you include in this table alongside your own molecule. So for example, as you're going through pharmacology studies, it's very common to use a reference molecule or an established competitor clone. And here you can provide that data all in one place. And so this is an example of a very handy way to keep track of that data. But how do we actually do this calculation? As a project team, you should agree on what the relevant parameter is for using your Mabel calculation. And usually it's one of the most sensitive parameters that is related to the mechanism of action. Because remember, the key is that you're trying to provide the safest starting dose that you can. So here, as an example, we'll insert an EC50 for B cell activation. Convert that EC50 to molarity, and during the pharmacology experiment, measure the receptor occupancy that occurs at these concentrations. So in order to calculate the Mabel, a simple Hill equation actually can work, and this has been explained in several FDA papers. So in this case, the receptor occupancy at this concentration is 15%, and plugging those parameters into the Hill equation and assuming a Hill coefficient of 1 in the absence of collected data that suggests otherwise, convert that to a first-in-human dose, in this case, 45 micrograms. And in this case, we're going to use a 70 kilogram human, but it is important to consider that your indication may not be a 70 kilogram human that is the most appropriate. And so at a mg per kg basis, we're looking at a 0.6 microgram per kilogram first-in-human dose based on this Mabel calculation. But you're not done there. You have to consider, is this a feasible dose to formulate and administer? And will this dose provide any clinical benefit? Because just because it's safe doesn't mean it's actually going to have any pharmacological impact. Another example of how to calculate a first-in-human dose using more of a modified approach is with receptor occupancy. In this case, you should establish a human PK projection from your most relevant species, such as non-human primate, and using the human PK projections, simulate the receptor occupancy established from your preclinical models. In this case, we're hoping to understand at what human pharmacokinetics we can result in a 30% receptor occupancy. And in this case, using the simulation, a dose of 0.01 mg per kg results in approximately 30% occupancy, and it decreases rapidly from there. And so this is just an example of how you might use this as a way to calculate a first-in-human dose. I'm going to hand it back off to Dr. Meyer.
Great, thanks, Aaron. So we've discussed starting dose selection, and hopefully now you have an appreciation for why this decision is so critically important and how complex it can be. Because the primary objective for a first-in-human study is to demonstrate safety. The first-in-human starting dose is a critical component of a clinical pharmacology development strategy. But dosing strategies for a phase 1 study of biologics often extend beyond just starting dose, particularly in oncology, where phase 1 studies are conducted in patient populations. So in addition to risk, an understanding of risk-benefit is required. So in addition to starting dose, other dosing considerations may include dosing interval, whether to use a weight-based or flat dose, what increases should be used for escalations, the targeted dose range based on an understanding of predicted optimal biologic dose or predicted plateau of effect, and a targeted maximum dose. And in addition to dosing, the clinical pharmacology strategy includes study design and analysis considerations. For example, the appropriateness of 3+3 design versus adaptive design, or the benefit of backfill or PK/PD cohorts at clear dose levels. Strategic sampling for PK, receptor occupancy, anti-drug antibody, and pharmacodynamics to acquire sufficient information to establish PK models and mechanistic biology-driven PK/PD models is very important in the clinical pharmacology plan. Interim cohort-by-cohort analyses and escalation and iterative population PK analyses and the associated data requirements are further considerations for your clinical pharmacology plan. And finally, exploratory exposure response or exposure safety evaluation, even starting as early as phase 1, where you're testing your drug in a patient population. And further, dosing recommendations are then made for recommended phase 2 dose and dosing interval. or for combination dosing, dose interval, sequencing, or duration of dosing. So now let's visualize an example. We have a moderate to high-risk compound for which we're using a Mabel starting dose calculation from an in vitro receptor occupancy assay that results in a starting dose of 0.03 mg per kg. We select a dosing interval of every 2 weeks based on a predicted human half-life of 15 days under conditions where target-mediated drug disposition is saturated. We select a weight-based dose due to unknowns in the target biology and dose-response relationship. We then select an escalation scheme of half-log increases, which is the standard, until we reach higher dose levels, where we then decrease the escalation steps, for example, from 10 to 20 mg per kg here. Based on translational predictions, we can predict the target effective concentration of 1 microgram per mil and a predicted optimal biologic dose from the human equivalent dose and PK predictions of 1 mg per kg every 2 weeks that may achieve this concentration in 95% of subjects. Then, based on a predicted plateau of PD effect, we can plan for a maximum dose of 20 mg per kg. We then support the clinical team in a 3+3 study design for this particular program, and we recommend PK/PD backfill of each cohort, up to 9 subjects per cohort, in order to further characterize the biology, since it's a novel mechanism. We could design intensive sampling at dose 1 and dose 6, the predicted steady state, and sparse sampling at all other doses. We also design sampling schemes for receptor occupancy, anti-drug antibody, and pharmacodynamics. We include PK-matched triplicate ECGs with intensive PK in order to characterize that risk. Our analysis plan will include cohort-by-cohort data visualization and non-compartmental analysis. We'll also include iterative modeling and simulation at flexible cutoffs depending on our data collection and speed of enrollment. We'll plan for exploratory PK/PD modeling to drive our dose selection and recommended phase 2 dose. But what PD or biomarkers should we model? Dr. Moss will explain more.
Thanks. Obviously, to progress through clinical development requires making a lot of important decisions, as you just saw. And often, you have to make these decisions in the absence of very clear-cut guidance. Biomarkers and pharmacodynamic readouts are what we want to use to inform those decisions. But this will only be possible if the questions that will be asked are considered ahead of time, earlier during preclinical development. Where you do not want to end up is being in a clinical program with a pile of biomarker or pharmacodynamic data that is gray, unclear, and having not considered what you are going to do with that data. So consider, what decisions will we have to make? What data are we going to use to inform those decisions? And then which specific biomarkers provide that data? And when will you observe changes in those biomarkers? And I don't necessarily mean in terms of time, but in terms of the dose escalation, what dose level is going to invoke biology or PD? And why are those biomarkers you selected relevant? If you can go through these questions and ask them before you ever even get to clinical study, you're going to be in a much better place. And this proactive approach will result in productive project team discussions around the expected data, how you expect the data to look, and why you expect that. This will force necessary project team introspection that will challenge your own knowledge and understanding and be very honest about what it is you expect to see and at what dose levels you expect to see it. One way I recommend, and that can be critical early in development, is to create another table, which may seem very simple, but at the project team level with many individuals who may not be experts in biomarkers or pharmacodynamics, this can be really helpful. And this is helpful not only for understanding which biomarkers, but also for developing your assay strategy. Because with a thorough understanding of your therapeutic comes the need to provide a robust and quantitative measurement that will inform those decisions. And so this is where this strategy comes into place. And what do I mean by strategy? And this table is a visual representation of that. And it provides a talking point for your project team and groups involved, not only with developing the assays, but the people who will be making the decisions based on that data as well. Christina?
Biomarkers are an important part of development, starting even before the first-in-human study start. And this continues throughout development, as I said, into the recommended phase 2 dose selection for complex biologics. So clinical pharmacodynamic biomarkers are really critical for establishing an understanding of the biology to drive this recommended phase 2 dose, particularly in the absence of other key endpoints like maximum tolerated dose. So in addition to PD, we also integrate clinical safety, efficacy, population PK, and relationships between all of the above to understand the totality of data to drive the recommended phase 2 dose decision. And for biologics, often we're dealing with complex scenarios. So how do we determine dose in these complex scenarios? For example, in the absence of maximum tolerated dose, as I mentioned, which is the case for many biologics. Here we really drive the dose selection using PK/PD and exposure response or exposure safety evaluation. In the absence of dose response or exposure response, for example, with anti-PD-1 agents above 2 to 3 mg per kg. In the absence of PK, for example, for complex vaccine formulations. For complex mixtures with several active components, like ADCs, how do we determine dose? In heterogeneous target populations, where the dose may be different, for example, in lung cancer versus melanoma. In synergistic combinations, where the doses may need to be reduced. Or in the absence of a true phase 2, which is becoming a more common scenario, where your study design may actually be intended as a phase 1B or 2 registration study. So ultimately, we evaluate whatever information is available, the totality of data, to try to understand drug behavior and patient response, to understand the quantitative relationship between all available parameters and the time course of all of those endpoints, to drive rational decisions around recommended phase 2 dose. So in summary, biologics are growing in complexity and many applications are currently in oncology. Key milestone tasks to be carefully considered in pre-IND early development include the target product profile, evaluating risk, which may have many layers, and selecting a first-in-human starting dose, which is more complex than for small molecules or simple biologics. The clinical pharmacology roadmap for complex biologics is centered around the predicted relationships between dose or PK and PD safety or response, and any of the above relationship with each other, and also identifying sources of population variability. In using this roadmap, we can drive drugs to success for marketing authorization and bring new drugs that can cure patients, like with cancer, to market. So with that, I'll close and answer any questions.
Thank you so much, Christina and Aaron. We'd now like to encourage all attendees to ask Drs. Moss and Mayer the questions that you have about this presentation. You can enter them into the Q&A box, and we will answer as many as we have time for. So it looks like we have our first question, and it's for Christina. Will regulators tell you if your starting dose is wrong?
That's a great question. So in my experience, the conversation with regulators around starting dose is definitely a two-way conversation. And if you submit, for example, a Noel-based approach with a very high starting dose for a compound that's high risk, where a Mabel dose will be more appropriate, the regulators will certainly recommend that. And they will also pull from their totality of knowledge about the drug landscape and target biology in order to guide sponsors around this decision. But I think it should be, if that is the case, it should be a red flag to make sure that you understand the biology. If you were going to recommend, a 3 log difference in starting dose than what the regulators were expecting. So yes, the regulators will work with you, but you really should establish a good understanding of your biology prior to having that conversation.
I have a question from our audience member for Aaron. Can you comment on your experience with assessing receptor occupancy in tumor biopsies from preclinical and clinical studies in oncology?
That is a really good question, and That is a very difficult data set to ask to obtain, given the complexity in using potentially tissues to detect receptor occupancy. Obviously, there's a lot of technical implications with how you might prepare those kinds of samples for receptor occupancy and the stability of those kinds of samples between collection and analysis. In my case, this has been something that has not been used for clinical application as much, just because of the inability to really confidently quantify that measurement, but it is something that I've seen in development a lot. And more often than not, a blood-based receptor occupancy will be used if possible, and other methods to try to ascertain tumor distribution and target cell binding. But tissue-based receptor occupancy can be very difficult without a lot of preclinical development and qualification to clinical samples. So I don't have a lot of experience using that for both, but there are opportunities to develop those assays.
Yeah, Erin, I wanted to add, there is some experience, I think, around anti-PD-1 agents having measured receptor occupancy in a tumor from a mouse model and relating that to concentrations and receptor occupancy on circulating T cells in the blood. Really, I think that didn't directly translate to the human, but it gave an idea of the relationship between blood and tumor receptor occupancy in order to drive an understanding of what the clinical meaning was of saturation of receptor occupancy in the blood, because that was the measurable endpoint, as you suggested. So I have seen that performed before, and it can provide utility, but perhaps it needs to be translated further, right?
I would just add also, thank you for that. I would add that we have a lot of modeling and simulation methods that we employ that have provided, I would say, pretty good guidance in terms of PBPK and mechanistic modeling to get a handle on what the tumor occupancy might be based on blood occupancy and other preclinical aspects. So we can use modeling and simulation to get at these kind of answers as well. That is just a very difficult assay to develop to robustly measure tumor-based occupancy reliably.
All right. Someone has a question for Christina, and Erin, feel free to chime in as well. Do you feel that pharmacodynamics drives your PK, your pharmacokinetic modeling and simulation strategy?
I think there are two really separate utilities. So one objective of characterization across all of clinical development is to understand the variability in your PK, independent of pharmacodynamics. So you want to understand what patient characteristics are leading to differences in clearance and other parameters. Then you also want to link that PK to the PD response, and that is ultimately what will help you make that decision for what dose is appropriate or whether you're even having a desired effect. So I think the utility of PD is not, the PD doesn't necessarily drive the PK modeling strategy. It's two separate efforts that go in parallel. Erin, do you have anything to add?
Oh, yeah, I agree completely, and I would add that they're sort of inseparable, so I don't know that it's necessarily one that drives the other, but more in tandem, the understanding of both. And there are times where the pharmacokinetics has to be implied from the pharmacodynamics in certain special cases. But in general, I believe they go hand in hand, and one doesn't necessarily drive the other, but instead they're developed together.
I've got a question for you, Erin. What do we do when the human target doesn't have a matching version in mouse?
This is a very common situation. I think as biologics become more and more complicated and the disease, the mechanism of action for the various diseases become more esoteric, it's harder to know how that translates in a preclinical model, and there may not be an instance where a direct homologue in a mouse exists even. And in these cases, I would go back to understanding the integration of all of the preclinical data and really everything you can do in the absence of an in vivo model that absolutely recapitulates the human situation. And that includes using your in vitro data and as much pharmacology as you can. So really what I would say is how well do you understand the mechanism of action? What can you measure in vitro systems, and how close can you get to the receptor in human? Sometimes there are orthologs or similar targets that can at least help you understand dose response or exposure response, even though the target may be slightly different, the pharmacology may still be the same. And then hopefully you can go through the process we talked about in terms of tox model justification. And in some cases, tox models aren't exactly similar as well. So really, it's a system of the totality of the data and integrating that data to provide the best argument you can that you understand the pharmacology and the relationship between how the pharmacology results in efficacy as well as toxicity.
Thanks, Aaron. We've got a question for Christina. How accurate would you say are your predictions for dose range?
That's a great question, and that really relies upon the translatability of your non-clinical data moving into first-in-human. I don't think there's been a comprehensive review of retrospective dose selection processes, so we really don't know. In my anecdotal experience, we do usually get pretty close, particularly when we're targeting the optimal biologic dose based on integration of data across several sources, in vitro data, different cell types, with an understanding of the distribution of that particular drug. Our predictability, I think, decreases when we get into more complex and novel platforms that don't have perhaps as predictable PK behavior as a monoclonal antibody therapeutic would, for example. So some of the smaller proteins or larger molecules, or when the PK/PD relationships are indirect and we don't understand that time course until we get into humans. So I think it's highly variable, and it depends a lot upon the properties of your compound.
Great. It looks like we have one final question, and it's for Aaron. When should we start preparing our first-in-human dose selection?
Oh, it's never too early in my mind, and incorporating some of the things that we talked about here at the inception of a project, I believe, is the best time. When you have a molecule or a therapeutic with a pharmacology that you start to get a sense will be beneficial in certain disease states, that's probably the best time to start considering what data am I going to need to select the starting dose. And really, as Christina, I think, really nicely described, that is not just where it stops. That is going to help define your clinical development. So as early as possible. And that will also help you define strategies for biomarker and pharmacodynamics. And these things have implications for CMC and formulation development. As well as, if you start thinking of these things that early, as soon as you have a project, when you have situations later, for example, not having a direct mouse version of a human target, for example, if you think of these things earlier, that might help you come up with solutions to those problems earlier as well. So I don't think there's a too early time. It's never too early.